A point mutation is a mutation that only affects a single nucleotide of a nucleic acid. They usually involve the substitution of one base for another, resulting in one of four consequences. Here we have the original DNA sequence, which has been translated into RNA. The red letters indicate the coding region. The codons from the coding region are then translated into these amino acids. I will illustrate the coding region using Skittles. In this mutant sequence, we observe a C base change to a G. This is known as a transversion because the pyrimidine base changes to a purine. A purine base changing to a pyrimidine is also known as a transversion. When we change the C base to a G base, this codon still codes for the same amino acid. This is known as a silent mutation. In this mutant sequence, we observe a G base change to an A. This is known as a transition because the purine base is changing to a different purine base. A pyrimidine base changing into another pyrimidine base is also known as a transition. When we change the G base to an A, this codon codes for a different amino acid. This is known as a missense mutation. In this mutant sequence, we observe an A base change into a T base. This is a transversion because it is a purine changing into a pyrimidine. Changing the A base to a T base in the DNA causes the RNA to transcribe a U base. This resulted in the codon coding for a stop codon instead of an amino acid. This is known as a nonsense mutation. In this mutant sequence, we observe a T base change into a C base. This is a transition because it is a pyrimidine changing into a different pyrimidine. Changing the T base to a C base causes the stop codon to code for an amino acid. This is known as a readthrough mutation, causing the continuation of translation beyond the normal stop signal.